It's now my honor to introduce a good friend, one of Boston's most successful and most innovative entrepreneurs, John Fish, the CEO and chairman of Suffolk, and who is also the board chair at Brigham and Women's Hospital. John is going to introduce the next panel. John.
Chris, thank you very much. Good morning. It's my pleasure to introduce the next speakers. The Novartis Institute for Biomedical Research, or NIBR, excels at collaborative innovation. Its mission is to develop technologies that can lead to therapeutic breakthroughs for patients, and it is a leader in the field. With its worldwide presence, NIBR brings together researchers who treat, cure, and possibly even eradicate some of the most devastating diseases and conditions seen in clinics each and every day. NIBR's president, Jay Bradner, often refers to drug discovery as a team sport. NIBR has cultivated an external network or teams of more than 300 academic and 100 industry alliances focused on areas of mutual scientific interest. The goal: to break down barriers in science and to accelerate medical discovery. Novartis has been a pioneer in cell and gene therapy, developing the first FDA-approved CAR T-cell therapy for cancer, and one of the first FDA-approved viral gene therapies. It continues to drive the development of therapies that can recognize and fight certain cancers, and it conducts research and develops technologies to prevent the treatment of genetic conditions of the central nervous system, the eye, and the blood. Every day, the research and early development teams at NIBR bring us one step closer to the world in which no target, no disease is undruggable. I am honored to introduce our next speaker, the president of NIBR, Dr. Jay Bradner. Dr. Bradner will be joined on stage by Meg Tirrell. Meg has joined us at the forum in the past, and we are fortunate to have her with us here again today. Meg is a senior health and science reporter for CNBC. Please join me in welcoming Dr. Bradner and Meg Tirrell. Thank you.
Well, thanks so much. I'm so excited to get to be here and to get to catch up with Jay. And I apologize if my internet might be going a little bit in and out. I hope that it's not. Jay, let's start by talking about sort of what the last year has been like for you through this pandemic. I was really struck by seeing some of the feedback from yesterday and the comment, especially from Peter Marks at the FDA, that gene therapy applications were flat in 2020, which really just sort of showed the strength of this space through the pandemic. What's it been like over at Novartis?
Well, thanks, Meg. And John, thanks so much for the invitation to join today in this panel. Always enjoy this event. Meg, during the pandemic, we worked hard to keep the labs open, both in response acutely with therapeutics potentially for COVID-19, but also with the idea that patients suffering from rare genetic diseases, sickle cell, pediatric acute lymphoblastic leukemia, that these children couldn't wait for a pandemic to end to have their definitive therapy, their cell and gene therapy reimagined. And this had some real organizational complexity, but the purpose-oriented, committed scientists at Nibr fought through the pandemic and all of its logistical and personal challenges, and we kept the lights on. 1,050 scientists badged in this morning at our Cambridge facility. And in order to keep them safe, and thankfully, knock on wood, we've not had any workplace transmission as yet. Many of us no longer at the bench are working remotely from home. We did, however, in the most acute wave of incident SARS-CoV-2 in April and May last year, take a focus of our portfolio, one of the largest in the industry, down to what we call the MEP, the minimal essential portfolio. Well represented on that list of programs were cell and gene therapy projects, because our portfolio is rapidly maturing to clinical investigation and we could prioritize that science. But moreover, because these medicines can be definitive and in some cases curative. And so even amidst this impossible global health crisis, we kept the lights on and the scientists badged in and kept innovating these cell and gene therapies.
Wow. And of course, the title of our chat is Collaboration is Key. And I wonder, through this experience of having to pare things back to just the essential, and also to figure out new ways to work together. I know we're going to talk a lot about working externally, but are there lessons that you can take from this past year and how you've had to change doing things that you think you'll carry forward that'll improve systems, in the future?
Meg, there are so many learnings from this pandemic about how we operationalize science. And in a way, the time away from the labs for some of us was a time of real reflection and strategic planning. We wrote a new strat plan during the pandemic, but also new ways of connecting to some organizations that we might have historically thought of as competitors and even treated as competitors. Everything from guidance from a small group of like-minded R&D leaders trying to create safe environments and share best practices for laboratories at difficult times, but also these interactions just naturally bred new collaborative interactions. I think in the space of cell and gene therapy, there's much discussion right now between the National Institutes of Health, Peter Marks at the FDA, Pfizer, ourselves, and others about what might we do in a more open posture to innovate platforms for cell and gene therapies for these low incident diseases that are maybe too small to grow large pharmaceutical companies like ours, maybe too complex for biotech industry to get all the way through to patient access. And these are actually getting pretty granular. And so we learned a lot about the practice of science, how it can be more flexible in location, but we also learned how vital it is to be shoulder to shoulder and innovating. And so I'm ready to get back to the labs.
I can imagine. That's so fascinating about getting together and with another giant organization like Pfizer and others, and trying to think about solving problems that it may not make sense for a giant organization from a business perspective to solve. I'm curious about the reasons that you see Novartis as the right company to be solving some of those problems that from Wall Street's perspective, those folks might say this is too small to matter. Why does it make sense for you as a business?
Well, it's a great question, Meg. We take these tough decisions all the time at the portfolio management boards that I chair with my partner, John Science Development. And it is true that there are some projects that are just not likely to be growth product yielding for a company of our size. And we're pretty disciplined about pursuing programs within the bulk of our portfolio that can really grow the business of Novartis. With that said, I think Novartis has a longstanding belief in this idea of access to medicines. And now with Vas, who's a global health clinician scientist, some of the work that we do and have done for years at the Novartis Institute for Tropical Diseases and malaria and cryptosporidiosis starts to read through into the space of cell and gene therapies. And you might have seen that we have an exciting partnership recently announced with the Gates Foundation to try to imagine a one and done in vivo cure for sickle cell disease so that the majority of patients affected by that disorder don't miss out on this moment of rarefied innovation just because they're in a challenging socioeconomic situation. Now, it can't be every small or narrow opportunity that we can organize around. And so the other way that we think that we're a good environment for those chances for patients is to be sort of like a rising tide. Some of the learnings in cell and gene therapy for us have been really hard. I know you've covered some of those challenges, and for sure, coming out the other end, there are paths within Novartis for patient access to Kymriah, CAR T cell therapies, Zolgensma, spinal muscular atrophy gene therapy, that have been a great learning as to how you bring such a complex medicine to patients around the world. And we find ourselves pretty happy to share that guidance in order to, like a rising tide, benefit this low incident tail of genetic diseases that might be well addressed by this technology.
How are collaborations changing? I mean, I've always sort of thought of what you do, your business, as a collaborative industry. I mean, pharmaceutical companies collaborate with biotechs, everybody collaborates with academia, but in many ways, you know, it's been sort of licensing it and taking it in, and then maybe the collaboration stops or changes. How is it changing now, and how do you see this kind of looking down the line?
Well, Meg, there's some highlights and there's some real lowlights that I'll share with you from just where I sit looking out at our ecosystem and reflecting on some of our most storied and successful collaborations, as with the University of Pennsylvania to innovate the first approved CAR T-cell therapy, but some of the more challenging interactions in the biotech sector as well. You know, it's just sort of an imperative. I mean, science today is totally non-disciplinary in the life sciences where therapeutics are concerned. Today's biologist uses, you know, command line scripting as a microscope, and the inventive steps that I, as a chemist, will take in small molecule lead optimization leverage, you know, augmented reality of machine learning and generative chemistry. I mean, I trained for 15 years to download the concepts and languages of chemistry, biology, data science, and medicine. It's just not feasible to go it alone, personally or institutionally. Innovation is so highly democratized today, and so geographically vastly distributed at baseline, but especially in cell and gene therapies, because molecular biology as a discovery tool is so accessible and inexpensive. A sophomore college biology major understands most of the principles essential to creating modular gene therapies. And so there's such a strong role for academia and biotech. Now, if you take that, this increasing translational and entrepreneurial nature of the academy, and you invoke the rocket fuel of today's accessible biotech investment, what is created is both exciting and challenging. It's exciting because there's an unprecedented resource going into discovery sciences, and this space in particular today. But it's challenging because much of that investment is directed towards fast follower, thinly differentiated, more redundant science, and very few out there beyond, you know, the great hype movie making declarations of founders. Very few is really reading through to disruptive therapeutics at proof of concept in the clinic. The third and last challenge I'll mention is, you know, this flood of investment into biotech is so exhilarating, but it has dramatically reset the expectations of investors and board members of these small biotech companies as to what it might cost, what it might rightly cost to access that technology for innovation. And in an environment where many technologies are exclusively licensed out of academic centers into singular biotech entities, we actually see slow progression of some therapeutic modalities to the patient. A good example is CRISPR versus targeted degradation. CRISPR came on the scene maybe 5 years earlier, but the challenge in accessing the freedom to operate, the sleeves rolled up, shoulder to shoulder collaborations in CRISPR have led to very few translations of CRISPR into the clinic. There surely will be more. In targeted protein degradation, where I've worked and innovated, by putting the design principles for these molecules out in the public domain with no extraordinary restrictions on access, there's dozens of targeted protein degraders entering human clinical investigation, even with small molecule chemistry being so much harder than molecular biology. In sum, I would say that there's some really golden opportunities to work together, but there are still some organizational and financial constraints that make it harder to collaborate, you know, without transformational partnership valuations.
That is so interesting. In terms of the CRISPR space, I know this is a focus that you guys have there. Tell me more about that. I mean, I'm imagining you're sort of getting at the patent issues. Is that right? And there's just been, I mean, this has been sort of like this epic battle from coast to coast in the patents with CRISPR. But how has that affected what you're trying to do?
Well, for Novartis, because we're a really well-resourced research organization, we actually are among the lucky ones that can access this estate of intellectual property and then build upon it with the actual therapeutic substances. It has required agreements with Caribou and with Intellia, and these have been very enabling collaborations above and beyond, access to foundational intellectual property. But they are barriers nonetheless. When new molecular biological modalities emerge, they're often less accessible, say, than chemistry innovations.
And why has it been different in the degradation space? Why has there not just been such a patent fight there?
Well, I'm not a patent attorney or strategist, and maybe there's one to come, I don't know. At the time I entered the protein degradation space, I was working at a cancer charity, the Dana-Farber Cancer Institute. We were tinkering with these open source models of science. They were sort of like social experiments with the accessibility to chemical probes. And when we solved a small molecule approach to targeted protein degradation with what we call phthalimide conjugation, you could think of it as sticking derivatives of Celgene's Revlimid onto targeting molecules to direct protein destruction machinery towards new targets, leading to the elimination of target proteins, not just their inhibition, which is what traditional molecules do. We wrote it up in a science paper. We put the reagents into the public domain. We filed intellectual property so as to enable therapeutics to emanate from the discrete targets of interest, but not in such a way, at least as it was intended, to block others from working in the space. In the fullness of time, I suppose we'll learn whether that reads through to the broad accessibility of this chemistry. But we already see a company, Arvinas, deploying cereblon conjugation chemistry and AR and ER degradation with first-in-class so-called PROTAC small molecule therapeutics. And it's exciting to see this science read through, to real investigational medicines.
And I guess I sort of wonder, what argument you would make for doing all of this in a sort of more open source way. I mean, just thinking about the incentives at play for the folks who are thinking about patenting something and the way in which they patent it. From where you sit, when you were there, you were at sort of, a charity. And so it made sense to do it the way you did it. Now you're at a pharmaceutical company that wants to access the technology. But for those folks who are, trying to make money off the technology,
yes.
What incentive is there for them other than just doing the right thing and making sure that these things are accessible to patients, which of course these scientists want to do? What argument do you make to them?
Well, what I encourage institutions in the academy and honestly in biotech and pharma is to create with some enabling technology platforms a mode of non-exclusive access. And the terms can be extraordinarily favorable. There can be access fees multiply derived to certain therapeutic technologies, and maybe the PD-1 patents will ultimately turn out to be one such example. But rather to invite multiple partners to leverage the platform, receiving then maybe even better ultimate financial outcomes because it's more shots on goal for the foundational academic institution. So I'm not suggesting that there should be a free ride into these technology platforms. These are valuable and hard-won discoveries, but I believe that it can be even more financially successful for the innovator and also for the institution through multiple non-exclusive licenses to parties that might use the foundational platform. Now, we're doing this at Novartis as well. We have one of the best lipid nanoparticle chemistry platforms. And we might have just, put this on the shelf for only our utility. But when genome engineering strategies have emerged, they're by and large ribonuclear protein complexes that need to be delivered. And one of the best ways to deliver these are with these lipid nanoparticles. You might know that these LNPs are a core component of the vaccines that contain mRNA that are so successful these days. There's been real innovation in the LNP field by us and others, and we are using our LNP technology within Nibr. But we've created, inspired by this idea of a more open framework, a pretty shovel-ready template for biotech companies to access these LNPs for their own use. And we have multiple partners now on that platform. I can't say that this solves all the challenges. And I relish the competitive nature of science. It's so good for patients. And the fact that there's real reward for innovators and institutions that solve historic challenges in the science of therapeutics is so rich today, is directing a lot of resource and innovation into the space. But I do think that there's a more open and collaborative posture that wouldn't sacrifice any of the rewards that especially academic institutions rightly expect.
Those are such interesting points. And when you mentioned lipid nanoparticles, it reminded me, and I just looked up my favorite Onion article of the entire pandemic, where the headline was, "Nation can't believe they spent so long overlooking obvious solution of mRNA instructions for spike protein encapsulated in lipid nanoparticle." So lipid nanoparticles are now part of the just sort of societal zeitgeist. Everybody knows what they are now because of the pandemic.
So. Oh, they're in everybody's bloodstream. And so...
So in our last couple minutes, I wanted to focus in on that point you made about how so much of the industry and all the money that's flowing in is focused on fast followers and redundant science. I thought that was such an interesting and apt term. What are you most excited about that you guys are working on in the cell and gene therapy space that is really potentially game-changing?
Thanks, Meg. It was about 5 years ago in our first strategic plan where our now CEO, then partner Vasner Simon, and I crafted a Nibr 2.0, a reconsideration of our scientific strategy that had enumerated what we called the new science of therapeutics. And in our disease areas of ophthalmology, neuroscience, oncology, and hematology, we see that the clarity of human genetics combined with these powerful new modalities can create really disruptive and potentially curative therapeutics for patients. And so even at a company the size of ours, we have to take focus because these are very expensive medicines to innovate and extremely expensive medicines to manufacture and provide access to around the world. And so we have a next generation now of CAR T-cell therapy that I'm very excited about, still directed at present towards blood cancers, but could be a platform for partnerships with all types of CAR T-cell innovators. We, as others are using genome engineering strategies like CRISPR to turn back on healthy fetal hemoglobin so as to compensate for the mutated hemoglobin in sickle cell disease. This medicine is now in clinical investigation. And then in gene therapy, we've been focused on adeno-associated virus 9 or AAV9 gene therapies, and continue to pursue definitive widespread therapy for spinal muscular atrophy, but now have 12 other neurologic and ophthalmologic diseases where we're developing gene therapies that go way beyond gene replacement, but now seek to deliver payloads that are themselves reimagined proteins, modular proteins that can short circuit disease. Cell and gene therapy can sometimes be about 90% of what we talk about externally, but it's still only about 10% of the portfolio. And the reason for this is we've learned through spinal muscular atrophy therapies, Zolgensma, and CAR T-cell therapy for leukemia, Kymriah, just how hard these medicines are, not to invent and to get to proof of concept, but to bring to patients around the world. And what we hope is that by building a global platform for the manufacture, the access, the value-based pricing, the distribution, and the covenants to regulatory authorities that persist after launch, that we're also a good partner for innovators who themselves are creating cell and gene therapies.
All right, Jay Bradner, this was fascinating. Thank you so much for this wonderful discussion. It was wonderful to get to catch up with you, and I know we'll get to continue the conversation. So thank you again. And to everybody else, I'm told by our organizers that we're taking a short break and everybody should come back at 10:50 a.m. Thanks again.
Thank you, Meg.